Okay. HIV is a retrovirus. That means that it contains RNA and an enzyme called reverse transcriptase. Reverse transcriptase enzyme is able to make RNA into double-stranded DNA. So what happens is the HIV binds to a receptor, CD4 I think it is, on the cell surface membrane of the host cell. And in humans, the host cell is the helper T cell, which you've heard about in the immune system. It's part of the cell-mediated immune response. So the virus binds to the cell surface receptors that are specific to the helper T cell. So the HIV virus cannot bind to any other cell type in your body, only those, because only the helper T cell expresses this receptor. Once it's bound, it's able to then inject the RNA into the host cell. The RNA then gets converted by this reverse transcriptase enzyme that also enters the host cell. That produces then DNA. So this is viral DNA. That viral DNA gets inserted into the host's DNA, and unfortunately for the host cell, it cannot tell the difference between its own DNA and the viral DNA. So once the viral DNA is then in there, the host will carry out transcription and translation using ribosomes to make viral proteins. Once you've got lots of viral proteins, they then are assembled to make lots of viruses. And when you get lots of viruses, they burst out, destroying the helper T cell. So the helper T cell is now unable to function, and you have viruses that are able to go and infect another healthy helper T cell. So at some point, the virus exists within the human body just as the DNA, and that's during the dormant period. So that's where you might have normal helper T cell counts, everything seems normal, you don't have any symptoms, you don't feel ill, and then suddenly the gene gets switched on, you make lots of viruses, those viruses then leave the cell, they coat themselves in a membrane from your phospholipid bilayer of your cells, and that helps again to disguise them from detection from other like macrophages that are in your immune system. They find another helper T cell, bind to the specific receptor, inject their RNA, insert their reverse transcriptase enzyme, make the DNA, which then gets put into another host's DNA, and that poor host cell then uses that viral DNA to make viral proteins, assemble the viral proteins into new viruses, and so the process continues. And that is a retrovirus HIV. If the HIV destroys enough of your helper T cells, so over time the number of helper T cells falls, then at some point there will be a critical level because the helper T cells roll. So this could be an exam question where they ask you, HIV destroys helper T cells, suggest the consequences of that. So you need to basically highlight what a helper T cell does. So a helper T cell releases cytokines. Those cytokines cause macrophages to come to the area to carry out phagocytosis. They also cause killer T cells or cytotoxic T cells to come to the site of infection and destroy infected cells, or also destroy pathogens. So there's a chemical called interferon, which is good at stopping viruses that are released by the killer T cells. And then the final one is how they activate B cells. So a B cell that has come across a pathogen is activated by these cytokines so that it will replicate by mitosis and then be able to differentiate, and that will then allow the B cell to make plasma B cells and antibodies, and those antibodies are then able to bind to the pathogen and destroy it. But if you don't have these cytokines because you don't have the helper T cells, if there are no helper T cells to make the cytokines because the HIV virus has destroyed it, then you're basically powerless against opportunistic infections. So you can end up developing flu, pneumonia, and other different diseases because you just cannot coordinate an immune response, because the main role of your helper T cell is to coordinate the immune response by calling in macrophages, killer T cells, and activating your B cells. you.